Today I will talk about my PhD project, which is on mineral weathering in presence and absence of microbes. So more specifically, it will be about the dissolution morphology in abiotic and biotic conditions. And I will try to propose some statistical techniques to possibly distinguish between one and the other based on surface microtopography. So first of all, why it could be important to study mineral weathering in the first place? Mineral weathering is involved in the geochemical cycle of many elements based on a series of dissolution and precipitation processes. Here, for instance, there is the weathering of silicates, which as a result produce a consumption of CO2. And so this is a natural sink of atmospheric CO2. And this is also used as geoengineering with the geoengineering techniques to capture CO2. So this is to give an idea of the possible implications that studying mineral weathering in general could have. And so now, when we study mineral weathering, the first thing that we want to know is the dissolution rate. The dissolution rate, in general, is defined as the amount of a given component that is released from a given mineral surface in a given time, and it is normally usually normalized by the surface area to have measurements that are comparable among different experiments. And we can measure this in two ways. There is one obvious method, which is by measuring the amount that is released from the mineral surface by actually measuring the increase in concentration in the solution. And the other way is actually a direct measurement. So basically, we are going to measure the variation in the volume that we observe at the mineral surface. And so we measure this by actually measuring the height difference between a non-reacted surface and the reacted area. So by measuring this height difference, then through the molar volume, we can get the dissolution rate. One important relationship, we have an important relationship between dissolution rate and the saturation state of the bulk solution under which the reaction takes place. So intuitively, here we are working in undersaturated conditions. So as the saturation state with respect, in this case, to calcite increases, the dissolution rate is lower. And on the other hand, if the saturation state is lower, then the dissolution rate is higher. And we have this specific relationship for calcite. Why is it important to have this kind of curves and to know the specific relationship that we have for a given mineral at given conditions? Because the indirect measurement of the dissolution rate is not always possible. This is possible only when the solution chemistry is actually informative. So when we are able to measure the variation in the aqueous solution, this is not always possible. So if we have this kind of curves, then if we measure the dissolution rate through direct measurement, then we can enter this graph, and by knowing the dissolution rate, then we can back estimate the saturation state with respect to calcite under which the reaction took place. But on the other hand, this is not always possible, because in order to have a direct measurement of the dissolution rate, we need to have a non-reacted reference, absolute reference at the mineral surface, and this is not always possible. So the question is, what can we say about the reacting conditions based on the surface microtopography when we don't know the dissolution rate? Can we say something? So this is where we start. We start from the qualitative observation, that the surface microtopography of a weathered mineral surface is different depending on the saturation state with respect to calcite in this case. So at very far from equilibrium, we have the generation of these so-called H pits. These are perfectly crystallographic H pits that in case of calcite have this rhomboedral shape. But as we get closer to equilibrium, this shape changes a little bit, and they become more triangular. And then as we overcome the critical value of the Gibbs free energy, for which the opening of H pits is no longer spontaneous thermodynamically, then we no longer observe these surface features, and the dissolution is more homogeneous along the surface. So we start from this qualitative observation, and the idea now is to find some tools to describe this evolution of the surface microtopography in a quantitative way. So the aim of this PhD project is to extract information about the history of a given mineral weathering that are left imprinted on the surface microtopography. And so to do so, since we want a quantitative information, the idea is to use surface roughness as a proxy of surface topography. So the idea is to find a link, a statistical relationship between a characterization of surface roughness after dissolution and the saturation state of the fluid under which the reaction took place. And then the idea would, once we find this link, the idea would be to find how the presence of microbes affects this link. So the first part of the project is related to the first question and will be addressed with abiotic experiments. The second part, we will add the biotic experiments. So let's start from the abiotic experiments. The setup is a flow-through system. So the idea is to have a flowing fluid which goes in and out the reactor in order to maintain within the reactor a constant saturation state. When we work in a closed system, if we are at undersaturation, we will have dissolution, but then we will reach at some point equilibrium because of the release of from the mineral itself. So we want a flow-through system in order to release the mineral that is dissolved in the solution. And then we continuously stir the solution inside the reactor in order to have as homogeneous as possible conditions inside the reactor. We are working with calcite, with natural calcite. So we started from a big natural calcite chunk. We cleaved it, and then we polished one surface in order to get rid of the cleavage steps, the steps that we have from cleavage, and also to have initial surfaces that are as comparable as possible among different experiments. We are working at ambient conditions, so ambient temperature, and we allow equilibration with atmospheric CO2. We are working in alkaline conditions, and then we change the saturation state in order to explore different saturation states. The total duration of the experiments is in between 8 and 11 days. And then the idea is that at regular time intervals during the experiments, the experiment was stopped. We removed the calcite sample from the reactor to acquire topography images of the reacting surface, and then we put back the calcite sample inside the reactor to continue the experiments. The VSI is a vertical scanning interferometry. Is an instrument for acquisition of topography images to give an idea of the scale. The vertical resolution of its sub-nanometer. The lateral resolution, so the length of a single pixel, is 200 nanometer. The advantage of this instrument is that it allows you to work with quite large field of views, which can go up to 1 millimeter. 1 millimeter for a single acquisition in stitching mode. So that is why it's pretty convenient for our purposes. So I will quickly go through our first study that we performed at a given saturation state, 0.6, so at close to equilibrium conditions where we don't have HPs. And what we notice is that starting from a relatively flat surface after polishing, we have, as soon as the surface gets in contact with the fluid, we have an evolution of the surface roughness. And then this surface roughness stops evolving. So we reach like a steady state surface configuration. So this is qualitative. And then the idea is to quantify this evolution with different techniques that I will not explain into the details. But it's important to point out that we perform this analysis on different windows across the mineral surface in order to take into account the spatial heterogeneity that we have on a given mineral surface. And so what we notice that the different statistical parameters reach a plateau that corresponds to the steady state configuration that we noticed on the surface. And so here the idea is that these specific plateau values could be related to the specific saturation state at which we were working. So based on the experimental data that we acquired, we developed a numerical model, which I will not explain in details, but basically this model was able to describe the statistical behavior that we noticed experimentally. And then we used it to try to predict the evolution of surface roughness at different saturation states. And what we noticed is that according to this model, we actually have a relationship between surface roughness and the saturation state of the fluid. So from this, the idea to keep looking for experimental evidence for this relationship. And so what we did is to perform these flow-through experiments at different saturation states, from 0 to 0.8. And what we can see here, just by looking at the topography, we can see that some of them are clearly distinguishable, also by naked eye. But for some of them, especially when we don't have clear surface features, is more difficult. So a quantification of this topography could be helpful to distinguish between them. And so I chose to show one of the statistical techniques that we use to distinguish between them. And this is the Semivariogram. Just to give you an idea of what is a variogram in general, is a measure of the degree of spatial correlation that we have from a given data set. So in our case, we are looking at the surface topography, so at the distribution of the heights value. And so the idea is that if we look at two points at a short distance, they tend to be more similar than points at a larger distance. So as we increase the distance, the data points become more and more dissimilar, and so until we reach a plateau value. So if you look at the variogram model that we use to fit the experimental data, we have a sill. We have a sill. The sill is the variogram plateau. So it's the value at which the variogram reaches a plateau. And then we have a range or a correlation length. And this value represents the distance, the separation distance at which the data are no longer correlated. So for distances larger than this range, than this correlation length, then the data are no longer spatially correlated. And so we calculated the semivariogram for all the saturation states, and we noticed a clear trend. So basically, we noticed that as the saturation state increases, the variogram goes lower and lower. So more specifically, the sill decreases, but also the range shifts to the left. And so we notice this trend for all saturation states. So the variogram keeps going down and down as the saturation state increases. And so if we plot these model parameters separately, we can see more easily the trend. So as omega increases, the correlation length decreases, and even more clearly, the sill decreases. This trend is clearly an exponential trend. This is a semi-log axis. And so how can we use this observation? If we have an unknown data set of a calcite surface that went through dissolution, but we don't know the conditions of dissolution, that we can measure the semivariogram and then enter these plots. And so once we know the correlation length and the sill, then we can back estimate the corresponding omega value and have an idea of the conditions under which we have dissolution. Of course, these values can be slightly different depending on the case, but at least we can have an interval of omega values. And so the idea is that under the investigative conditions, we are able actually to back estimate the reaction conditions by simply measuring the surface microtopography after dissolution. So it's important to specify that this is under the investigative conditions. So this is true when we are working at the same pH, the same temperature, but also could be very important also that we need to work with mechanically polished calcite samples, because this, as long as we are dealing with close to equilibrium conditions, where we can still clearly see the polishing scratches, this could have an impact in this relationship that we see. But as long as we work with mechanically polished calcite samples, there are other possible applications of this technique. So once we develop this relationship, then we could develop a similar relationship, but a different relationship by adding one by one a given chemical to the solution to derive another expression, and then by comparing these two relationships to isolate the contribution given to the surface roughness given by a specific chemical. And the idea is to do the same also with microbes. So developing the same kind of relationships in the presence of microbes, and then by comparing the two relationships, isolate the contribution given by life to the surface morphology, so to the dissolution morphology. And this is what we are going to do in the second part of the PhD project. So this second part of the project will try to address this question, whether or not the bioweathering results in specific imprints at the mineral surface that could be unequivocally attributable to the action of life. Here, the keyword is unequivocally, because more often than not, surface features that have been attributed to the action of living organisms, very often they could be reproduced through purely inorganic processes. And so it is difficult to distinguish between them. So just to give you an idea of the kind of ambiguity, for instance, these filaments that were dated 3.5 billion years ago, they were interpreted as resulting from the action of living organisms, based on the similarity with the modern cyanobacteria. But on the other hand, we can produce similar filaments by using purely inorganic processes. And so only based on morphology is difficult to distinguish between surface features or in general morphology that are resulting from life or non-life. So to give you some more examples more related to weathering, here we have some examples of surface features that resemble in size and shape the size and shape of bacteria, but these are produced through abiotic processes. But on the other hand, here we also have some surface features that have been interpreted with other techniques as resulting from the action of living organisms. And so the idea is that probably if we want to use surface microtopography to distinguish between alteration, abiotic alteration and bioalteration, maybe we need to develop some new techniques that are able to distinguish between these two. So here I would like to go through quickly through all the interactions that we can expect between a crystal surface and the presence of microbes, for what we know from the literature. One effect that we can observe is the enhancing of weathering, the enhancing of dissolution. How? Basically, from a decrease of pH. This decrease of pH can occur locally, so at the interface between mineral and microbes, or more globally, so by changing the pH of dissolution itself. And this could lead to the observation of specific features that we wouldn't see in abiotic conditions. We could observe H pits at saturation state where we wouldn't expect to see H pits, and overall an increase in the dissolution rate because of the decrease of the saturation state. On the other hand, we can have inhibition of weathering. How? Through inhibition of reactive sites. So basically, if we have the presence of bacteria on the surface, they will somehow cover the surface. And so by covering the reactive sites, maybe they could inhibit the formation of H pits, or in general limit the dissolution. Or for some bacteria, we could have an increase of pH. And so in both cases, we could have an inhibition of the growth of H pits, and overall, we expect a decrease in the dissolution rate. Lastly, there are some for some specific bacteria. There are some specific bacteria that are able to actively bore into minerals, and so for this kind of surface features that we can see are some microtunnels inside the mineral substrate. So I will show you some examples of this specific interaction. So for instance, here we have the effect of Shewanella on calcite dissolution. Shewanella is not supposed to do anything with metabolically with calcite, and in fact, their impact is actually an inhibition of calcite dissolution. So here we can see that in abiotic conditions we have H-pits and we have dissolution, whereas in biotic conditions, the presence of Shewanella strains on the surface stops dissolution, so we don't see H-pits. And also, if we compare the reference surface with the reactive surface, we don't see any surface retreat. We don't see any high difference. So we don't have dissolution in presence of this strain. On the other hand, there are some studies that show that some specific strains of cyanobacteria are able to actively bore into calcium carbonate substrates, despite the oversaturation with respect to calcite, aragonite, and all calcium carbonate minerals in dissolution. And so a different mechanism could be involved. In this other study, we see that a specific cyanobacteria strains, a filamentous true boring strain, is able to bore into a calcite substrate very deeply, so for more than 100 microns. And so they, in this study, Garcia, Pichel et al., they propose these mechanisms where basically they uptake calcium at the boring front, so they will locally decrease the saturation state, and so they will make dissolution possible thermodynamically, and so they will keep going down and down and down with this dissolution mechanism, and at some point they will stop when they will be in so much depth that they will not see enough light to survive, and so at some point they will stop. So our idea is to maintain our experimental setup, and maintain a flow-through system. So probably we will slightly change the aqueous solution, but the idea is to keep having a flow-through system so that we can have a constant saturation state inside the reactor, even if with the presence of bacteria inside will be much more difficult because of their metabolic activity. And so we will still work with calcite to take advantage of the work that we did in the abiotic conditions. And regarding the microbial strain, we decided to start trying with this strain, PCC 7203, which is a cyanobacteria strain. And so since we are using a cyanobacteria, we will need to add light to the experimental setup. Why the choice of this strain? Because this strain they showed that they are able to form intracellularly amorphous calcium carbonate inclusions. So they are able to uptake calcium from this solution. So the idea is that maybe by uptaking calcium, they could do something similar to the mechanism that we noticed for the filamentous cyanobacteria strain. And so maybe this will tend locally to decrease the saturation state. And also, these are suspected to be endoliths. And so something that we may want to check if they actually actively bore into carbonates, or they just live inside the holes and crevices that there are at the surface. So this is our first start, and we are currently doing some tests to check this hypothesis. And so, what is the expected impact of this strain on the calcite dissolution and on the dissolution morphology? So, as I said, one possible effect could be the sequestration of calcium, which will lead to decrease the activity of calcium. But on the other hand, when we provide light, they will do photosynthesis. So the CO2 consumption will act on the carbonate bicarbonate conversion. And so in case of CO2 consumption, we will have an increase of the activity of carbonate. And so it is actually impossible to predict a priori the impact of the saturation state with respect to calcite. But on the other hand, if we switch off the light, then we would have both trends going in the same direction. And so we would expect a decrease in saturation state, with respect to calcite. So, and also one idea could be to have light darkness cycles, because their impact on the surface is not necessarily something that is reversible. So it's possible that with these cycles that they would lead specific imprints at the surface. So from the first test that we did some batch tests to see if they stick at the calcite surface. And so what we noticed is that they tend to create a biofilm at the surface. So another thing that we need to consider here is also that once they create a biofilm, we could have a different chemistry at the interface. We could have partial or total passivation because of their presence. And also they could leave specific imprints due to their local mechanisms. So in conclusion, these are some SEM pictures that we just took yesterday. So here with the sample prep, it was with critical point drying. So in order to try to maintain the shape of the bacteria, and we noticed that after only 22 hours in batch reactors, they were already able to stick at the surface, and so this makes us confident that flow-through systems can be possible, because so it seems that their attachment rate is quite high. And another thing that we notice here is that we have huge precipitation. You actually have calcite growth. So it's possible that we don't have yet the analysis of the chemistry of the solution, but we can expect that in this specific case, the pH increased so much that the activity of carbonate is much higher than the variation in the activity of calcium. And so this is why we see calcite growth. So this is work in progress. And just to conclude, the idea overall of this work is to develop some statistical techniques that are able to distinguish quantitatively between weathered surfaces and bioweathered surfaces, based on the acquisition of surface microtopography data after dissolution. So before concluding, I would like to thank all the people that participated and that are participating to this work, to all the laboratories that where I'm working, and a huge thanks also to the biomin group, which taught me basically everything I know about cyanobacteria. Thank you again for the opportunity to share my work, and I'm happy to take your questions.
Thank you very much, Luca. So if anyone has a question, is in the chat on person, you can ask them. If it's in the chat, maybe raise your hand and Juliette can make the interface between the question in the chat and one of in person. So does anyone has a question right now? I have several one actually. So I have a first more question at the very beginning of your talk. When we were talking about the roughness, the fact that the surface of your bits, okay, yeah, this one. When the surface of your calcite stabilizes, does it mean that the dissolution stops, or is it just has reached an equilibrium between dissolution and deposition of calcite? Because between this state and this stage, is there still dissolution happening, or it's just a stop?
Yeah, no, actually there is still dissolution happening. So if you look at this surface, so if you have absolute reference, so when your dissolution starts, this surface keeps going down, but just the surface roughness doesn't increase anymore. So yeah, but it keeps going down with time.
I'm just trying to understand your experimental setup. So if you can go to the slide 7.
Yeah.
So you said you showed in the later you did some experiments with different saturation. But here you say it was an open system with a pH of 8. So did you manage to buffer the system to keep it at pH 8, or there was no control on pH?
Okay. Yeah.
What was the chemical manipulation to give it pH?
Yeah. Okay. So we were working with very high concentration of sodium. Okay. So the composition of the solution was sodium chloride, sodium hydrogen carbonate to regulate the pH, and calcium chloride. So basically, the sodium chloride concentration was very high, and so the idea is that we could change the saturation state with respect to calcite by maintaining quite the same pH, by only changing the concentration of calcium. So basically, we were only changing the concentration of calcium chloride. Okay, and by doing so, the pH was always there. It wasn't changing.
So then the later question was to the biotic setup, and I guess we had some point of discussion. Because in the biotic setup, as you said, pH is not uncontrolled because of the microbes itself. Is it possible that the mechanism we showed about how the surface topography is affected by the microbes, instead of doing it on a calcite mineral surface, if you do it on another mineral surface, which is not that sensitive to the carbonate activity, just to study if there is an impact of the microbes?
Yes. Yeah. I guess
It's just a very broad
yeah,
question.
it's I mean you have infinite possibilities of
Sure.
choices, and
I guess it's related to the point that how generic is this idea of like surface topography can be sort of an indicator of microbial activity. And if it's a generic idea, can we test a different system which is not that sensitive to the photographic setting?
yeah, that's an idea, but I mean, my hope is that with a flow-through system, we are able to. I mean, okay, so maybe the idea is to. Okay, if we go back to the. Okay, here. So the idea is that if we are able to inject very low concentration of cells inside the reactor at the beginning, and if they attach very quickly to the surface, and then if we can have a very high flow rate, but with the cells still remaining attached to the surface, then I hope that we will not see too much change in the chemistry of the solution. But the only difference that we will see will be local difference, and so that is actually what we want to see, the local impact of the microbes, despite of the overall conditions in the solution. So that is what we will try to see. Then if it is not possible for a calcite, we will see. But yeah, that's the idea.
I don't see many other minerals that will not be sensitive to pH. And that's basically, if you put protons in that, you're gonna protonate things and these things. So I mean, you can silica, silicate, or whatever, it will be sensitive to it.
Not for pH, but it is more about the carbonate.
Yeah, for carbon.
It goes with the pH, right?
In the abiotic system, it's a nice.
I mean, one way, but maybe so another thing. I mean, if it's about the carbonates, I mean, you may wonder what's going on if you start decreasing the amount of inorganic carbon.
Yeah.
And this is what you're going to play on to decrease the saturation, but you would also,
yes.
I mean, I don't know, maybe those guys, there will be like some function with the activity of the microbes.
Yeah.
You know, they may also decrease there, but there's something that will go on there that is kind of, I don't know, maybe predictable, but I just don't catch it.
So I have a question in the one question which is related to that one, I think.
Yeah.
To get your cells growing, do you have some phosphate, or what is your medium you have in your system? In that case, you can have some maybe a diet kind of thing. I don't know if it's ready. But you say that I didn't see the culture medium you.
Yeah, because we are testing different things. So we started by using BG11, so the medium that we use for optimal growth of the bacterial strains. But then in that case, you could have precipitation of many mineral phases, and so maybe it's not the best idea. So what we tried in this last test, where from which we get these images, is to just work with a very simple medium with just sodium nitrate and sodium bicarbonate, and just adding a drop of a very concentrated with cells very highly concentrated to the solution. And we noticed that after 1 day, they managed to attach, to stick to the surface. We don't know if they were still alive or not. So it's
pH 10, so I guess they are not so bad.
Okay.
Otherwise, you would not move the pH.
Yeah.
In my opinion, I mean, if you don't put the microbe you don't see the increase of pH. So if it increased, it's due to the microbes. And so
yeah,
like they can do that in this kind of solution, right?
Yeah. But maybe they increased pH and then they died. But
I think that something which can be really interesting is about the number of cells that you have at the beginning. If you want to see some processes, because you are dealing with two things, opposite things at the same time, you have dissolution and probably some precipitation if you don't have any pH which is stable. So to be sure what's going on and what's about at least a few number of cells can be easier, and to see some local stuff. Growing either we have to be growing condition or not. It's something else from my point of view. At the beginning, we want to see what can happen about metabolism, what's going to be the interaction between the cell and the material. So I guess that you can try perhaps two things with a low number of cells and a dense culture, just to see what's going on and what are the differences.
I also have another question related to biotic part. Since you have biofilms that can develop on the surface of your minerals, and that you are trying to look at the surface itself, do you plan to remove the cells from medium, and how do you plan to do that?
yes, okay. So we will do different things. So yeah, of course, it's difficult to look at the biofilm and then to remove it with the same sample, because also for the sample preparation that you have, you need to carbon coat it, coating the surface, and so once you do that, you're no longer able to then remove the coating and the cells and looking at this topography. So probably we will have to do that with different samples. So on one end, we want to see the biofilm itself, so to look at the cells. But on the other hand, we also want to then remove all the cells after at the end of the experiment, and then to look at the topography itself and to see what is their impact, what are their signatures, and
yeah, do you know? Brian, do you know a way to remove the cells without altering the surface? Do you know there's an easy way to do that? Because
yeah, I'm not sure. For what I know that there are some organic, something that dissolves the organics without altering the.
So what I got from Damien is also that
Yeah,
with VSI, you may be able to still look at the topography without removing anything.
yeah, that's the other possibility, but I'm not sure with carbon coating. But,
We have a question from François.
yeah.
No, actually, this was not a question. Thank you, Luca. This was more a comment about this last point about organic matter and trying to retrieve the crystal surface. In principle, you know, these so-called cold oxygen plasma techniques, they should not, well, that's for sure that they will remove all the organics because they will convert it to CO2, even the carbon coating and everything, this will be gone. And in principle, these techniques, they should not damage the surface. But well, they are sold for this. But of course, it's yeah. That said, it's probably needed to test it, right? It's and it may take more time. But in principle, this cold oxygen plasma, cold oxidative plasma, they are very efficient. Sometimes assisted with UVs or not, but in general, it's just a cold plasma machine. I used it, you know, when, for instance, when you want to analyze mineral fibers in the atmosphere, for instance, and you want to separate them from organic fibers, which are dominant, it's really very, very efficient. But well, yeah, then you have really to be sure that it's not damaging anything in your surface, because your purpose is really to, of course, if the cold plasma is moving the topography, then you have lost everything. So probably you would need to test things before, after, without organics, and put the organics. But techniques exist, definitely.
Yeah. Thank you very much.
Yeah.
Mm.
What kind of controls are you using for the biotic settings? Like, for instance, for this one, do you know how much is the pH increase without the calcite chip? So, like this, if I understood correctly, this is from the setup where you have your calcite with the microbes in the growth media. Is there another control where you know how the pH increases when the cells are just growing in that particular growth media
Okay.
recipe which you are using?
No, we didn't do that yet, but yeah, I think it could be useful to know. But I don't think it would. I would expect the same increase.
Because you're changing the recipe, the growth media, because now this is in sodium nitrate and sodium bicarbonate. Only.
Yes.
Whereas the regular standard way of doing this is in BG11, which has different things. So I'm just curious how the pH increases in just.
That you can answer.
Yeah.
You have the one in BG11.
Yes. Yeah. Okay. Yeah. Actually, in BG11, the final pH was 9.5, whereas in the simple medium with cells, the final pH was 10.
And this was without the calcite chip.
No, both with calcite chip. Yeah.
So far, the goal was to stick a cell
For now.
on the calcite chip, so that was really the only goal.
I'm just
Because it's
curious.
a bit complex, and I think, as Luca was mentioning, it won't be the same when you start flowing something through that.
Yeah.
I'm just curious because just to see how the pH variation, because at some point it can go too high, and if the calcite dissolution is happening, if there was some precipitation.
It should, yeah. I mean, you should get some equilibrium at some point with the
But at
calcite.
what point is the
Yeah. And another thing I was thinking of is, I mean, would be cool to, I think that there could be a way maybe to coat a calcite chip with an EPS only.
Okay.
No, and then you could try again your experiments at different saturation or undersaturation states, and try to see if you keep the same topography, the same relationship, and you will not have cells, you will have EPS only covering everything, and so if the EPS basically binds on the reactive side,
Yeah.
you may expect that I don't know, you change either the threshold for HPs.
Yeah,
That could be cool if there was a way to, I don't know, spin. Have you done that? That's like.
I think I saw some stuff like this to fix bacteria on, in Stixen, for instance, with the walls. They use some organic material and retrieve it.
PMMA,
So like the terms, yeah, because you do, then you will have the.
and you can speak code then.
Stick to that.
Yeah.
I'm not trying with EPS.
Yeah, and we could try maybe because there are some strains that produce a huge amount of EPS. And I don't know if you can spin code that on a calcite chip or maybe not, right? But
yeah, that would be very interesting. Yeah.
I just have a question. Did you try to check for carbonate inside of the cells, these cells, because as they are close to calcite, do they enrich in calcium content? Because this strain is special. Not all the cells have carbonates inside. So my point is, can we see, at least from the chemistry first, and of course by imaging, if there is some enrichment of calcium inside the cell? Did you try in X in this cell? That's not
no, it was, of course.
You're on that side.
Yeah, so we thought about this, but we are sitting on calcite, so we are not,
I know, but I don't know.
we have not tried too long. And even, I mean, we were on calcite, and these guys have been through water plus ethanol, critical point drying. So we started very quickly to look for inclusions. But then that was the thing. We thought, okay, we should have collected some suspension, put it on the filter, and check whether they had the inclusions, more or less, and so on. So probably this should be on the list for the next experiment.
To sample a part of that just before all the protocol for drying.
Yeah, but still, with that idea, if you take them from the suspension, then maybe those cells.
No, but maybe we will be able to see the inclusions, but at least not going through critical point drying. This is why I realized that they look empty, but then you would expect that they lose their inclusions when you go through that procedure. I think so.
And just the cells which are sticking to the surface, I mean, what do you think it is? Like, is it going to, what is the mechanism by testing? It's just the shears, or they are going to, if you have quite a high shear rate, they will just leave? Because you're thinking of high fluid in your flow setup. So what do you think it is? Like,
No, it's, we'll see what will be the best flow rate, but it's 0.25 milliliter per minute.
No, I guess, but do you have a sense of like how are they sticky?
Yeah, so it's from these fibers, but I don't know about
the fibers are.
But like, is it strong enough?
I mean, those things. I mean, you know.
Well, we did.
You bat them in
several bats,
positions, including ethanol and so on, and eventually you have that.
You know, agitation.
Yeah.
Really stick,
So, yeah, for sure we lost some of them. Yeah, for sure we lost some of them because at the beginning the surface was green and then.
This is what induced after 22 hours.
Yeah, this is.
I think usually the people, the way they describe the formation of the biofilms, the first interaction is electrostatic, and then you start binding with the EPS. So, and I think those fibers that are sticking, I mean, they are mainly like that. We also saw the same stuff in the aquaria on some diatoms. We could see diatoms sticking with those fibers on the surfaces and attaching to it. In my opinion, and maybe the difference between several strains is that maybe some are I don't know, maybe they produce more of this and then they stick better than others. Because this one is sticking, but I'm not sure that all the strains are sticking as well.
Even the culture, in the spectrum class, some of them seems to be really sticking to the plastic or the glass. Other strains.
Because in that, if this is a mechanism, somewhat, then maybe growing them in a stress-free condition would be better, so that they are producing a lot of EPS and just stick to the surface. Maybe they won't stay alive for long.
Yeah, there might be conditions where they stick more.
Maybe if it's so the PG11 modified recipe would have the minimum.
I mean, maybe this is not reproducible, but Luca, the last experiment, the PG11 versus this one, PG11, there was nothing left on the calcite. So maybe the calcite was flipped, I don't know. There was no cell on the calcite, and this one, there were many.
Yeah.
I think there's a question in fact. Just curious, do you plan to dope your calcite with some iron, for instance, to force the microbes to really dig and pick up some iron, which is the dissolution? We can do this kind of, maybe not iron, but this kind of experiments that will force the dissolution.
Yeah, that would be. Yeah, I think I don't plan to do it in this during the PhD, but that would be interesting, I think. And yeah. Yeah, also it's natural calcite, so maybe there will be traces of metals, and so maybe that it's a good reason for them to dig.
Yeah, this has been one big argument for digging into feldspar by some microbes that they will go for phosphorus.
So this strain seems to need a lot of calcium. But perhaps the media without calcium may need more the calcite too.
Okay, is there any more questions? Or maybe we can call it a day because it's been 1 hour. So thank you very much for attending again. Thank you, Luca, because it's really interesting, I guess. We've understood what you've done. It's quite exciting. So see you, I think the next seminar will be in 2 weeks. There was something that I never need to check, but this is so yeah, we will receive email to remind you about where and when to present. And thank you again and see you soon.
Thank you.
Thank you.
Hi, thank you.